Biological Sciences and Bioengineering Department of IIT Kanpur is certainly among the best in the country. On one hand, the department conducts top-notch fundamental research, albeit aimed at understanding the mechanism of different diseases. And on the other hand, there are bioengineering colleagues who develop interventions, clinical interventions, either from the perspective of tissue engineering or from the perspective of gene therapy. This department publishes in some of the most competitive journals where fundamental research is published, and at the same time has garnered one of the largest industry-funded project for preparing gene therapy vectors, which will be used for animal testing and soon for clinical testing, hopefully. The department has state-of-the-art central facilities for microscopy, for biochemistry, for radiation analysis, and all those. The department has access to a state-of-the-art animal facility, and IIT Kanpur is also the home for the National Cryo-EM facility, which is used heavily by colleagues from the BSB department. And our biggest strength is our fantastic students, whether it is the BTechs or the MTechs or the PhD students. They are certainly among the best in the country.
My lab is a developmental neurobiology lab. In very simple terms, what that means is that we study how the brain actually develops and gets its final shape and form. So there are many developmental disorders, one of which is called holoprosencephaly. So we are using the chicken and the mouse embryo to understand how holoprosencephaly occurs. In holoprosencephaly, the midline of the brain does not bend inwards, and as a result, the brain remains as a single chamber, and the two cerebral hemispheres that we have, they cannot form. So our lab has made certain fundamental discoveries in some of the genes and molecules which are involved in this process, and thus we have found out how embryos develop this developmental disorder. The other aspect of my lab also studies neurodevelopmental disorder called spinal muscular atrophy, which is a genetic disorder. It's a rare disorder. So we are looking into developing indigenous therapeutics which are available in India for the population of people that are affected by this devastating disorder.
We are the lab of neurosystems, and we are working in primarily two areas. The first one is understanding sensory processing in the brain. So there are many steps that happen in this, and we try to understand these steps using simple model organisms like flies and mosquitoes. We are working particularly on the sense of smell, and our goal is to understand how does a smell become attractive or aversive to an animal. We are recording the activities of different layers of neurons using electrophysiology or calcium imaging, and we also perform behavioral experiments and computational modeling to understand how these simple brains are able to process this information and learn something in this process about the general principles of brain functioning, which will also be applicable to humans. The second area that we work on is developing software tools for behavioral problems like depression and anxiety. We believe that if computerized solutions can be used that can provide effective therapy without the involvement of an expert, then we can take these solutions to much larger population. These can be scalable and cost-effective solutions. We have created a tool called Treadwell that provides automated psychotherapy based on the principles of cognitive behavioral therapy and has been shown to have effectiveness in treating depression in people.
We use fluorescence, bright field and confocal microscopy techniques in our lab. These make sure that we capture high quality images, which we later analyze for our research purposes. These are mouse embryo, which is another model organism that we use in our lab to study development of limbs and brain. We also use Cree-Lox-Pre system and in utero electroporation for gene manipulation in mouse.
Our lab generally focuses on developing an advanced microscopy technique, also known as super resolution storm microscopy. The full form of storm is stochastic optical reconstruction microscopy. We generally use advanced set of microscopy, that is the storm microscopy, which can resolve the structures to 20 nanometer scale. it has these two cameras that are cooled generally for having a proper visual structure. Here is the image of microtubules that we can see. Our lab generally focuses on resolving the subcellular structures for more detailed understanding of the processes that are going on inside the cell.